Right. Hi, everyone. Thank you for joining our webinar today. Today, our Technical Specialist Officer, Dr. Irene Liza, she'll be talking about how you can maximize your sequencing success rate when you're working with qPCR or short PCR product. So I think let's begin, and I'll pass the floor over to Irene. And by the way, my name is Rose. I am the International Sales Manager for Apical Scientific. It's nice to know everyone here. All right, Irene, so I pass this over to you.
Thank you so much, Rose. Yeah, all right. So my name is Irene. I'm the application specialist and a certified trainer for Apical Scientific. So today I'll be talking on the maximizing your sequencing success rate using short PCR products and also qPCR products. So let me introduce you to the scope of presentation for today. So first of all, I'll be doing an introduction on tail primers, and then I will move in specifically to M13 tail primers. We will look into how M13 helps in short PCR fragments as well as qPCR, some important FAQ, sample requirement, and I will close with conclusion. So what is tail? Tail means thermal asymptomatic interlaced. So this is the term that is used to sequence, basically sequences to sequence and analyze unknown DNA fragments that are adjacent to the known sequence. So this means that you have a known fragment and you have an unknown fragment that is linked to a known fragment. So basically, this particular tail primers was used as a selective enrichment, but then later adapted into numerous applications. For example, used for restriction sites for cloning, universal primers for sequencing, as well as some of these sequences were used for in vitro transcription. So basically, tail primers were used to facilitate downstream application, whereby they use non-specific utility sequence, again added to the 5' terminus primer. So these are some of the features of the tail primers. So if you see, you have an amplicon here, and you have usually primers, they are very much specific to the sequences of the amplicon. So you have a specific sequence that these are the primers, and attached to the primer are the tail primer. So basically these tail primers are non-specific sequences. So the tail primers are usually non-specific, and they are actually synthesized together with the sequence specific primers, which are also known as your target primers. So we know that the specific sequence primer, which is the target primer, they are very much specific or complementary to the vector sequence, which is the amplicon. So the melting temperature for this particular primer is approximately about 58 to 63 degrees Celsius. On the other hand, for tail primer, the tail primers are relatively short. So basically, they are approximately about 8 to 17 base pair long. So due to that, they are having a lower melting temperature, which is approximately about 47 to 48 degrees Celsius. So let's look into what are the advantages of having tailing primers. Now let's look into simplicity. Now when it comes to tailing primer, it is basically simple and convenient because it reduces or it makes the workflow much more easier. Then we have the high specificity, because with tailing primers, the proportion of non-specific products, they are literally very low. High efficiency. Tailing primers basically can yield to very specific products, and we look into this in coming slides. Speed. Basically, when you have tailing primers, now you do not have to work on cloning and transformation. That literally takes you more than a week. So literally, when you're using tailing primers, it shortens the whole workflow itself. Less risk. When it comes to cloning, usually what happens is it involves ligation step. So when there's a ligation step, it actually increases the chimeric artifacts. So when we are using tailing primers, now literally it reduces because now the primers are ligated with the tailing that makes it more specific. High sensitivity, so literally any single copy sequence basically can be amplified using the tail PCR primers. Now there are few types of tail primers. Okay, so the first one is basically random primers that which is we call this as either 6-mer or 9-mer deoxy ribonucleotides. So these particular sequences are very much common when we want to build cDNA. So we have a capturing area. So we tend to use random primers to capture. Then for M13 primers, so these primers, they are used in sequencing because now you can actually target the sequencing inserts, which will be the topic that we'll be discussing today. We also have linkers as another type of tail primers. So these linkers actually consist of restriction sites. So these restriction sites are added to the 5' of the primers because later on we want to clone them and we want to digest, and perhaps we want to subclone them into another vector. So these linkers work for subcloning purposes. Now let's look into into details of M13 tailed primers. So what is actually M13? So M13 is basically sequences, which is approximately 17 long sequences that is actually attached to the 5' of the primer sequence. So basically your forward primer, like what I've said earlier on, it is very specific to the target gene or the amplicons. So M13 is just going to be at the upper part of it. So primers are basically synthesized. These whole set primers will be synthesized, used in amplification, and finally in sequencing. Now, what are the common problems faced by researchers? Now, usually researchers will come across high divergent targets in microorganisms that they tend to lack in a universal conserved region. So because of that, they have to use degenerate bases so that they can capture most observed genetic variation. So when they do so, there can be mismatches between primer and target sequence. So literally that gives a serious impact on amplification efficiency. Now, this makes more worse if the template concentration is low or there are inhibitors that are present, especially when it involves a diagnostic sample. So how does M13 actually solves it? So M13 able to improve the robustness of amplification of PCR, inclusive of qPCR as well, and also improve overall performances of the amplification process, can neutralize the destabilizing effect of mismatches from the template or the target primer. And now you can use also more primer variants in a PCR or qPCR reaction. On the other hand, also increases the binding affinity, whereby it also reduces the possibility of error. Now, let me give you an example. So this is basically the black color highlighted is basically the amplicon. So we know that for sequencing, basically you can sequence full with just one primer. So what happens is when you are sequencing using forward and reverse, and basically you actually can get an overlap sequence, yeah. But what happens here is when we use a normal primer, which means the amplified primer for sequencing, all right, what happens here is we tend to have noise in the beginning. So literally you tend to lose at least about 20 to 30 base pair in the beginning. Then only the sequence improves. So what we do to obtain the first part of the sequence, we align the sequences with the reverse primer perhaps, or the vice versa, and we try to obtain the first part of this particular sequence. Now, what happens if you have in this particular sequence, you tend to have mutation, or perhaps there could be some QE bases that are very low. So you wouldn't know whether it is due to mutation or you wouldn't know it's because of sequencing error, right? Because from here to here, you can see an overlap. Basically, you can get a sequence to see whether is it a true mutation or sequencing error. But on the other hand, here You can't see that, right? So all these can be overcome using M13. So you just have to tag in an M13. So now literally the lost sequences will be M13 and not the sequence of your own. So what are the general limitation when it comes to short fragments? So short fragments basically involving PCR or qPCR less than 150, we see a lot of noise in the beginning, and you tend to lose a lot of sequences in the beginning. And when it comes to viruses, okay, so it has a higher variability region. So for you to obtain a full coverage, you have to do degenerate primers. So that's not only specific for virus, but could also be done for other microorganisms as well. So these primers that we are doing, I mean, basically the degenerate primers, they are only suitable for amplification purpose. So how M13 tail helps in Sanger sequencing? So M13 tail in Sanger sequencing, once you use it, you can actually obtain full sequence of the short fragment. Why? Because now when the noise involves, it only involves from the M13 side and not taking the sequences from the primer sites, okay, of your particular target gene. And now you can also able to tabulate regional mutation, especially when it comes for disease screening using qPCR or PCR technique. Easy to use because it is a very simplified workflow from our side, and from sample up to the answer, the workflow can be completed in 5 days compared to cloning that literally takes you more than a week. So sequencing M13 primer, what do you actually get? You get a greatly increased short sequence quality. So even though they are short and previously using the normal primers, there are a lot of noises. But when it comes to M13 now, it improved the quality because the noises are absorbed by the M13 primer. It gives an accurate sequences, full coverage of the target sequence, and also eliminates the needs to do cloning. Let's move on into M13 primers specifically for PCR products. So for our services, basically for M13 tail service, so we are going to use a 17 base pair length sequence adjacent to the 5' primer. So this is the primers and that is the length. Okay, so what type of PCR fragments can we use? So basically any fragment size that is less than 150 base pair. And if you are studying a microsatellite studies, then you can use an SSR or STR markers. So SSR is short sequence repeat and STR is short tandem repeats. And if you are doing some screening for disease genes, you can also use M13 tail primers. So what is the workflow? So basically, all you have to do is upon finishing your PCR, you submit to us the unpurified PCR samples only. So from our side, so upon raising the samples from you, so in our facility, what we will do is we will synthesize the M-tail primers. Okay, we will synthesize the particular primers and then we will run a PCR, we will do the cleanup, and then we will use M13 primers to capture the sequence through Sanger sequencing and finally perform codon align. So basically upon optimization, the turnaround time for the whole process is approximately 5 days. So what are the information do you need? Do we need basically for us to do this particular workflow? So what we need is we need the primer sequence information, both forward and reverse, that is actually used in the PCR amplification. Second, we also need a reference sequence. So this reference sequence is something that you use to amplify, basically where you have generated the primers. So the reason why we want to look into the reference sequence is because later on, using codon aligner, we will get to see if there's we will do this particular analysis and to see if there's mutation or perhaps any deletion, insertion. So we get to see all these particular analysis in that. Also, we hope that you could submit unpurified PCR product only. So we only accept unpurified PCR product only, and to fill up the biohazard form. So basically, this is the sample submission form. So on the top part, you will see the biosafety declaration form. So this form is basically to declare that your samples are safe and free from any hazards. Okay, so for PCR product, basically you have to just run gel, and this particular band should be visible on agarose gel using 2 microliter. Also make sure the size is a little more than 90 base pair with the minimum volume of 15 microliter. So this is the order type. You can click into the appropriate order for your particular analysis, name of the gene inserted. Also share the sequences of forward and reverse primer as well as type of samples. So these are the extra information for the DNA template. So you can fill up as many samples as you have. So basically, what is our workflow? So for all optimization, which means for the first time when you are getting the service with us, we will take at least 2 weeks inclusive to order and also to synthesize the M13 Primer. So this M13 Primer will be synthesized with your target gene. Right? So for this particular process, it takes about 2 weeks, but subsequent order, the turnaround time will be about 5 days, which includes the quantification of DNA sample, M13 PCR amplification. We will also have the PCR product purification, forward and reverse Sanger sequencing using the primers, and finally we will align with the reference gene with the codon aligner to provide you with the information on the mutation, insertion, or deletion. Let's move on to qPCR products. So for qPCR product, we are still going to use 17 base pair length sequence. So we still have the sequences similar as to what we have done to PCR. So, and for type of fragments, it is the qPCR fragments, because generally all qPCR fragments are less than 200 base pair. So if you wish to sequence them, you can purchase this particular service. So the workflow is still remain the same. You are still we will still only accept unpurified qPCR samples. So any samples containing primer or probes, primer and intercalating dyes, for example, like cybergreen, you can submit as they are. So we will do the purification in our facility. So again, the workflow is still the same. We still do the PCR cleanup, Sanger sequencing, and codon aligner. And the turnaround time is approximately about 5 days upon optimization. Now, the good news is we also cater M13 tail services for COVID-19 samples as well. So we also have synthesized the primers for M13 tail services. So we have the primers according to WHO, which uses the RDRP gene as well as E gene. Also for CDC protocol that utilizes N gene. So these are the primers that is already been synthesized. All right, we have it ready in our facility. So if you are going to send us the sample, so it will directly move into the PCR. So we don't have to do the PCR optimization that takes 2 weeks. So it will directly move into PCR purification, and you will get your results in 5 days time. All right. So besides COVID-19 samples, so what kind of other fragments that you can submit? So as for troubleshooting, say for example, if you have some bands that is coming in your NTC or negative control, so you want to know what is the what causes the contamination, you can also now sequence using M13 tail primers. And for example, if you would like to study disease screening, genetic diversity studying, study population structure on plant or animal, you can still use M13 sequencing. You want to identify insertion, deletion, mutation, also can be done through Sanger sequencing with our M13 tail service. So what type of sample do we need? So we still need information for the primers, both forward and reverse. We also need the reference sequence, which should be covering the flanking sites to submit. we only accept unpurified qPCR products, and also finally for you all to fill up the biohazard forms. So there's also here exactly the same. The declaration form is on the above. You can just click them down. And then for qPCR, the specification should be CT value less than 33, size more than 90 with a minimum volume of 15 microliter. So this is the order. So you can check the box according to the services that you want to purchase. Also, don't forget to let us know the name of the gene, forward and reverse primer sequences, as well as to attach the reference sequence. So this is the additional names for, you can fill up for your genes that involves that you would like to use this particular service for. Workflow is still the same. So only for the first time, except for COVID-19 samples, yeah. So only for the first time, all optimization needs, it takes about 2 weeks because we'll be ordering and synthesizing the primer. So subsequent order, it will take about 5 days only. So in these days, in this particular time, we will do all the quantification, amplification, purification, sequencing, and finally aligning it with the codon aligner. So what are some important questions that sometimes we get from our customers are, can M13 tail primer use for multiple species sample? So M13 primer is not used for sorting purposes, so therefore it is not suitable for environmental samples or multiplex reaction. So basically what we have is, as long as you can get, it is a pure sample, it is a single intact band, yes, you can submit for this particular service. So if you have primer, probes, and dyes, will it also affect this service? This will not affect because you will be sending the unpurified samples. So from our side, what we will do is we will purify the samples before even proceeding to PCR and so forth and so on. So conclusion for this particular sharing is M13 Tailed Primer Amplified Products gives a better sequencing result. The workflow is literally very simple and you can obtain the results within 5 days upon optimization. You can use this particular sequence for other analyses, basically if you want to do a community identification, mutation screening, or others. So this particular service will be an excellent choice to replace cloning and transformation because it actually saves the time and also the cost. Now we also have a good news because we are doing an introductory promotion with 20% off for these particular services. So you can get information from either our sales manager or from your distributor regarding these services. So thank you for your attention.